Thank you all for joining us for another Technion Australia webinar. In fact, this is webinar number 20 that we're running, and we appreciate anyone who's been following us. And if it's your first time, then welcome. We hope you enjoy this. I wanted to firstly welcome our guest speaker today, Professor Andrew Levy from Israel, from the Technion. Andrew will be formally introduced by your moderator today, Ruth Ratner, but I just wanted to thank you personally. I know you're a busy man, to giving up your time and sharing your insight and knowledge and expertise. For those of you who don't know me, my name is Ori Danieli. I'm the CEO of Technion Australia, which is the representative arm of the Technion in Australia. The Technion is Israel's Institute of Technology and among the world's leading dedicated to science, technology, and engineering. In Australia, we're part of a global network with a mission to support our scientists at the Technion by providing philanthropic support as well as scholarships and funding for infrastructure. We also facilitate research collaboration and partnerships with local institutions here in Australia. So without further delay, I'm very pleased to introduce Dr. Ruth Ratner, who has been involved with Technion Australia for over 12 years. She is the president of the local society, and she is a GP from Sydney. She also teaches medicine at Sydney University, and she is an author as well, who she might want to say a word or two about that if that's interesting to people. And I'm going to leave you now and join back towards the end. I hope you enjoy it. As with all our webinars, you're encouraged to ask questions. I'm sure everyone knows how this works by now. Click on the Q&A button, and Andrew will do his best to answer. So on that note, Ruth, over to you. Thank you.
Thank you, Ori. The kind of author I am is the one that has a very big bottom drawer. So thank you for the nod to that. And thank you, Professor Andrew Levy, who's agreed to give us a talk on a topic that I think affects an awful lot of people. I titled it From Sorcery to Magic, because for me, I think it really has proceeded through that for many years. And what I think Andrew's doing to me is magic. From the beginning of time, humans have been suspicious of the other, appearance, behavior, customs, beliefs, anything that wasn't the same as us. In any community, those differences were, and maybe still are, regarded with either fear or awe, or possibly both at once. Unable to comprehend what caused people to be different, outside influences were blamed. The raspberry that a mother ate, the gods that were not appeased, the neighbour who cursed a child, or the spirits that left a changeling in the place of a beloved baby. Thankfully, knowledge brought some level of understanding that dispelled those primitive notions, and differences in behaviour acquired labels and categories. Modern science identified chemicals and hormones that cause certain behaviours at both a whole person and a cellular level. But what drove those changes remained a mystery until advanced imaging was able to demonstrate in which organ exactly where an event was occurring. And still we didn't know what was driving the abnormalities, or the normalities for that matter. Observation showed us that certain diseases or traits run in families. Attention deficit disorder is a clear example. Whether they knew it or not, there's always at least one parent of a child who can admit to having similar difficulties to their child, often proven by surreptitiously testing out their child's medication. Of course, the huge breakthrough of last century was the study of genetics and the mapping of the human genome. With the aid of powerful computer programs, untold numbers of genes can be identified and compared across millions of people, throwing up the one genetic abnormality that only occurs in someone with a particular disease or characteristic, and ultimately the unraveling of that gene and the potential to alter it, from a curse to a cure. And Andrew is leading the charge in that in a particular area of neuroscience. Professor Levy completed his molecular biophysics and biochemistry degree at Yale, and then went to Johns Hopkins School of Medicine, where he received a combined MD, PhD degrees. He then completed an internship and residency at Johns Hopkins Hospital in internal medicine. After completing his residency, he moved to Harvard Medical School, where he received fellowship training in cardiovascular disease. In 1998, he moved to Israel, made Aliyah, and joined the Technion, which has been his home ever since. He's currently a professor at the Faculty of Medicine at the Technion IIT. The primary goal of the Levy lab is to find new therapies for children with various disorders using personalized precision medicine and patient-derived disease models. Professor Levy will discuss work from his lab demonstrating proof of concept for this approach in a child with a genetic disorder which has resulted in severe drug-resistant epilepsy, autism spectrum disorder, and severe intellectual disability. Andrew, I hand it over to you to explain where you're up to.
Thank you very much, Ruth. I really appreciate the opportunity to talk to all of you, and greetings from Israel to all of you, to all of you. So as Ruth mentioned, what we're going to have as our focus today, and I'm going to use this as a general, this can be a general roadmap, if you will, for approaching many disorders that we thought were really not been able to really consider treatment in the past. We're going to talk about neurodevelopmental disorders today, primarily. And what we're talking about in this case is affecting as many as 2 or 3 out of 100 children. These are often inclusive of intellectual disability. We commonly measure this in terms of a child's intelligence quotient. We're talking about children with IQs in the order of 30 to 35, severely disabled, not able to perform anything independently. Epilepsy, as many as 30 or 40% of these children, drugs are totally ineffective for their epilepsy. And autism spectrum disorder, which is a very heterogeneous disorder. Many of the children with severe neurodevelopmental delay cannot speak at all, have very limited social interactions. Taken together, these often come together as a cluster of symptoms. Very debilitating, of course, for the families, and difficult for the children. Many of these children, particularly with the epilepsy, may have up to 100 or 200 seizures a day. Very difficult problems, and as I mentioned to this point, and as Ruth mentioned in her introduction, very limited approaches to really try to help these children. But that's what we're going to try to focus on today. How do we try to help these children and their families? I want to emphasize that all of these disorders that I mentioned, intellectual disability, autism spectrum disorder, epilepsy, are very, very heterogeneous. But we're going to use that to our advantage. We're going to use that to our advantage because that's been one of the, I think, difficulties in approaching many diseases in medicine. And we're really at a major turning point in how we approach trying to help people with different types of disorders, whether it's breast cancer, whether it's intellectual disability, whether it's epilepsy. We're taking a much more personalized approach, and I'm going to spend a little bit of time talking about that because many of you may have not heard about this concept, this idea of personalized medicine. I want to say just a word or two about my own motivation for entering this area. It's on a very personal basis. Have decided to devote a major portion of my laboratory to try to understand a particular neurodevelopmental disorder, again, for personal reasons in my own family. Originally a disorder that was affecting very few children, and now we know affects about 500 children. So both as a scientist as well as a parent, in this case as a grandparent, very much familiar with this disorder. Okay. There are going to be a number of questions asked during this session, and we'll have definitely periods of time to discuss. I want to try to address all the questions that you have, and Ruth will help me to moderate that at particular times. Ruth, please interrupt me if you think it's an appropriate time to ask some of the questions that the group is asking. So I'd like to start with this concept of personalized medicine. You may hear about this and use different terminology, precision or personalized medicine. What do we really mean by that? Let me use an example that many of you are perhaps even more familiar with than the neurodevelopmental disorders we're talking about today, breast cancer. We know breast cancer, maybe 1 out of 7 in most studies, all women are going to develop breast cancer. To date, until very recently, it's been the approach to helping people with breast cancer has been the same, regardless of what a person's background may be, genetic background, or the cause for the breast cancer. The treatment is often very aggressive, and even though we know as many as 40% of the women with breast cancer do not need such aggressive treatment. When we talk about personalized or precision medicine, we take that particular disease and based on the fundamental understanding of that patient's disease or disorder, we then try to design therapies. Again, this is a major shift in medicine. We often group patients together with cancer, with liver disease, but now, because of the opening up of the human genome, we can understand the basic mechanism of what's going on, why a particular patient will develop breast cancer, why a particular child has severe intellectual disability. This is the starting point for developing a personalized approach to how we can try to help these children, to help people with any disease. This is, again, across the board. And this is going to be a revolution. This is already beginning, a revolution in medicine, which is going to dramatically improve care, reduce side effects, complications, and giving drugs to patients and people that don't need the drugs, and giving more potent drugs that are designed on the fundamental underpinnings of why that particular person has a disorder. Now, it's not been possible really to approach many of the disorders. I want to use the example of ASD. I know because this is a very difficult topic. As I mentioned before, very heterogeneous condition. Many children, adults with ASD, extremely high functioning. What we're focusing on today are children that are not high functioning, that do have ASD. Nonetheless, ASD is an extremely heterogeneous disorder. And the way to approach this, again, is to develop specific understanding of what is responsible in that particular child or adult for what might explain why he or she has this particular problem. And we're going to talk about how we go about doing that, designing specific models using the patient's own genetic information to try to understand what is the problem, what is the reason for this particular disorder, and then how we go about trying to help that person to overcome this particular handicap or disorder. Okay. I use the word roadmap here because I think that it's a roadmap that is applicable to virtually any disorder. And so the tools that have become available to us in the last few years, I want to just briefly outline for you how we might approach this for a child with, let's start again with a neurodevelopmental disorder. So let's use as a simple example, we have a child who, let's say he's 8 months old. He's not sitting up yet. He's not saying any babbling yet. He may be having seizures at this time. He doesn't make any eye contact. Usually that's a little bit later, but doesn't make any eye contact with his parents or caregivers. How do we go about trying to understand if there might be a genetic underpinning to what's going on here today? We know today that we've identified many genes that are what we call, there's a genetic mutation, a change in the DNA sequence that is responsible for these different disorders. How do we identify that? How do we figure out if that might be happening? So what's done today is that we take a small bit of blood, usually about a teaspoon, from both parents and from the child, and we do something called exomic sequencing. This is essentially sequencing or determining the sequence of the DNA for specific portions of the DNA in the parents as well as in the child. Some of you may have studied what DNA is as in high school or in college or learned on your own. But just for those of you to bring us all up to the same page, DNA we can think of sort of like beads on a string. And that's the way that we determine the order of those beads. There's basically 4 types of beads. We refer to them as by letters A, G, T, or C. And a particular gene, on average, is made up of about 1,000 beads. Okay? These beads eventually are translated into protein, and that's what's responsible for the particular property of that gene, whether it's for eye color or hair color, or functioning in the brain to function some kind like a channel that allows ions in. But this is the fundamental dogma of how our genes affect the structure of who we are. It doesn't affect in many different ways. So again, we have the capacity today to determine the order of these beads on the string. And we can do this virtually for the entire human genome. Each of us has about 30,000 genes, and we can do this very quickly, within a matter of days. And even in some places, even within a day, we can determine this for anybody. And this is done in a very automated fashion and very reproducible. And we can identify the sequence, as I said, in the parents and in the child. And we can identify if there's been a change that occurs in the child that would, let's say, predict that that particular gene would be damaged by this particular change. We can read the beads on the string, and they give us a lot of information about what might have happened to that particular gene. We call a change that is causing disease or disorder a mutation. And this is very, as I said, very straightforward today. And for this particular child, this is exactly what we did, presenting again at about 8 months of age with these particular handicaps. And
if I can interrupt for 1 second there, you were also then able to identify children with similar symptoms worldwide with the same genetic problem.
That's right. So this particular disorder we'll be talking about today, you know, all these disorders, neurodevelopmental disease knows no ethnic or geographic boundaries. Some of the work I'll be talking about today, we've been successful in finding some treatments which we hope will help to improve quality of life of these children. I'm in contact with parents from China, Korea, United States, England. There's parents also in Australia that have the same disorder. As I said, there's no boundaries. And this is true that, you know, I hope that, you know, this is science and medicine. Again, we can move across our prejudices and our own little worlds, and it builds a lot of bridges that are extremely touching and important. And I've really enjoyed working with parents from all over the world that we all share a similar hope and goal.
We just had a question. Is that the same as chromosome testing?
So, okay, a very good question. So our DNA is located in chromosomes. So everybody here that we're talking with today has 46 chromosomes. And we call them, they're usually in pairs. So we have a chromosome number 1 up to chromosome number 23. The 23rd chromosome is what we call the sex chromosome. So women have 2 X chromosomes and a man has an X and a Y chromosome. Remarkably, all humans are 99.9% identical in their DNA sequence. The very small 0.1% is what's responsible for what, might cause us to have slight differences in how we look, our tone of our voice, things like that. But we're so remarkably similar, which I think is also an important message for humanity. We often focus on our differences, but similarities. The mutations that we're talking about today, of course, are just can be as much as little as one bead on that string is changed. That can be the whole difference in what happens with a particular disorder. And the one that we're talking about today, again, one bead out of 1,000 in 1 out of 30,000 genes has been changed. And this results in a child having this combined severe disorder. And this is really the case with virtually all of these neurodevelopmental disorders. Again, as I said, affecting about 2 or 3 out of 100 children. Very frequently, we're finding, again, a single bead that is out of place or his spelling is incorrect. And this is what's responsible for the disorder. Okay. Okay. So I think what I'm going to talk about now is once we've figured out what is responsible for which gene might be responsible for or is changed in the child, but might not be present in the parents. So let's think about this for a minute. It's very frequent that the child may have a change that is not present in either of the parents. This makes it a little bit easier for us to, if we will, convince ourselves at this point that this is an interesting and important gene and trying to determine if that's responsible for why the child has this severe neurodevelopmental delay. Sometimes both parents might contribute one copy, what we refer to as recessive copy of the gene, and then the child will have both copies and have this particular problem. The many genes that are responsible for severe developmental delay are on the X chromosome. If you recall what I said earlier, women have 2 copies, men only have 1 copy of an X chromosome. So what we see is that the majority of these neurodevelopmental delay children are actually males, because it only requires that be 1 change in 1 X chromosome, and the male child will have the particular disorder. So many of the disorders are actually on the X chromosome, and many of them are not present in either parent, and we say is a de novo mutation that is responsible for the disorder.
Andrew, if a female has 2 X chromosomes, do they not have twice the chance of having it?
Very good question. Most of these disorders are what we refer to as recessive. This is a concept that, again, I want to try to explain. Many of you might be familiar with, but just to bring us all up to the same page. If we have for every single gene, we have 2 copies. If normally, of course, they're normal. If only one of those copies is abnormal, most of the time, we're able to function quite well, because the mutation is what we refer to as a loss of function. And the other copy can more than make up for this loss of function in 1 copy. And this is why most females do okay with mutations in one of their X chromosomes. The male with only 1 X chromosome, of course, doesn't have any way to make it up. The Y chromosome is lacking this information. And this is the case this is the problem with these sex chromosome linked disorders. For other disorders, though, you're right. If it's in a chromosome number 5, both males and female may only have 1 copy. And so unbeknownst to many of us, we may carry 1 bad copy, but 1 good copy, but we're okay. It's only when the couple gets together and by chance, both of them on chromosome number 5 at this particular gene, they have a mutation. 1 in 4 of their children might have a disorder of this nature. And many of you in the audience may be familiar with this genetic screening that is possible to undergo for potential parents to try to determine for example, for Tay-Sachs and cystic fibrosis today, whether this might be something to worry about for prospective parents. Okay. All right, so we identify a change in the beads on the screen in these children. Okay, so does that mean that that's what's responsible for the disorder? And what's so important about that? Well, first, let me answer the second question so you'll understand why we want to spend so much time in this. If we can identify the gene that's responsible for this, we can then go on to build models in the laboratory to try to develop therapies to try to improve the outcomes for these children. But how do we know if this is the right mutation? Maybe it was just by chance. Many changes in the genome are not deleterious. So how do we know? How do we know if this is a bad mutation? So the first thing that we do is we look to see if there are other people, other children with similar mutations. So as I said, out of 1,000 beads, there may be other children that have a very similar change in a very similar location in the same gene that have very similar symptoms. This, of course, helps to reinforce or helps to convince us that this is a potential smoking gun responsible for the particular disorder. But really the most important evidence comes then from trying to look at the gene and see how it changes how the gene functions. I talked about before, so the gene is a piece of DNA. The DNA is then, the cell converts that into protein. What is protein? Our hair is protein. Our eyes are made out of protein. Our skin is made, our body basically, obviously contains a lot of water and minerals, but the structure, most of the structure that you're looking at when you look in the mirror is protein. And that's what the genes code for. So do we want to ask, is that gene that has this mutation, has it changed its function? And how do we do that? And has that change in function, can that predict the kind of changes that we're seeing in the child? So we take 3 different approaches that are mutually synergistic, that go together. One is that we can actually take that piece of DNA, that gene, from the child and actually put it into bacteria or viruses, not coronavirus, but any virus, a different virus, often like chickenpox virus. And we can make large amounts of that protein. And we can study it in a test tube to see how its function has changed, what its function is, and to see how its function has changed. This is, again, a very, I say routine, but a very standard approach that is possible today to take any gene and to make large amounts of that molecule and to study its function in the laboratory in a very isolated way. So this gives us a handle on what the function and how the mutation has changed the function of that particular gene. 2 other models, though, that are quite revolutionary. We're building on the work of others that have, the first that won the Nobel Prize just 2 years ago in a part of medicine called stem cell medicine. So the second is to make models of the disease using a technology called Crispr, which again won the Nobel Prize, I think 3 years ago. And these are remarkable technologies which allow us to now create models of any single disease, any disease that we want. If we have a mutation in a gene, we can now create a human model of that particular disorder. I want you to just think about this for a second. Ruth was talking about, what was sorcery before. We didn't understand, we treated all children with intellectual disability exactly the same. We really had nothing to offer. Children with drug-resistant epilepsy, we throw, literally these children are having the kitchen sink thrown at them. Many of them have tried 20, 30 drugs with severe side effects. They don't work because we don't know what we're treating. We're just trying to do things empirically. So we can take this gene that has a mutation in one of its beads, and now we can use these 2 models. let me talk about the first model, stem cells. What is a stem cell? A stem cell is a cell that has the possibility of turning into any other type of cell, okay? So if I have a stem cell from one of you, I can then make that stem cell turn into a brain cell, a tooth, an eye cell, a heart cell, a kidney cell, anything that I want. It's really quite remarkable. We can direct what we call the differentiation of that cell into anything that we want. But how do we get that stem cell? This was the revolutionary breakthrough. From a hair follicle or from a small piece of skin, from a drop of blood, I can convert any cell to become a stem cell by sticking in 4 other genes into that cell. I can make it now become what we call pluripotent. It can now differentiate into any cell that I want to. So in this case, we take a child with this particular disorder, and we can take a piece of skin, let's say, or a hair follicle. Obviously, one hair follicle actually, and we can actually then the cells that grow in the hair follicle, we can introduce these 4 genes. DNA gets into the cell very easily. We have a number of technologies, very simple to enter the DNA into the cells. And the cell now becomes a stem cell. I can then treat the cell with a variety of substances, for example, extracts from the brain or extracts from the heart, that cause it to become any cell that I want. In this case, we wanted it to become a brain cell, and a particular type of brain cell called the hippocampal neuron. We have different markers that we can look at, different markers in these cells. We say, this indeed now is a hippocampal neuron, because this is the kind of cell, way it looks, the kind of proteins it makes as the hippocampal neurons in our normal brain. So remarkably, what we have now from a child with this disorder, we can make neurons from him or her that then we can use to study his disease in a petri dish. And get ahead of myself a little bit, but what we're able to do then is to study the biology of his neurons, the actual child's neurons, in this petri dish. And it sounds almost like science fiction, but these cells actually have properties that are similar to what happened in our brain. These cells build bridges between one another, which is really the anatomical basis for learning. What learning is essentially is when one neuron speaks to another neuron, and they build connections, is what's called the synapse, the connection between one neuron and another. And we can study this in the child's own neurons. As it turns out, this is what's really we're understanding is the fundamental basis for what causes many of these neurodevelopmental disorders, this problem with the communication between one neuron and another. In this particular disorder, the cells also have a propensity to undergo spontaneous discharges. Use a fancy word there, but this is again, for a cell, the correlate of what epilepsy looks like. So these cells spontaneously start to undergo discharges similar to what happens in the brain of a child that has severe epilepsy. So this then is a good model for us and helps again to reinforce that we believe we found the right gene that we can study in a petri dish to try to understand the pathophysiology. The second technology I want to talk about, and this is that we build a platform for designing disease, is called Crispr. This is a great example of how, things in biology, we never know where they're going to go. What something that's found, in this case, it was found again in some primitive bacteria that had this capacity for when a type of virus that would invade the bacteria had the ability to modify the DNA of that particular virus. Okay, so if you're interested in saving lots of bacteria, you'd want to stop that, but Probably not. In any event, they learned how to harness this ability to change the DNA sequence specifically at any point that we want, to now be able to do that for in animals, and not yet, but coming soon for many diseases, although being used already, I should say that, for human diseases already. CRISPR is a technology that allows us to edit the human genome, or the genome of any organism that we want to, at any spot that we want. I mentioned before there were 30,000 genes. We have literally billions of letters, billions of these beads. We can now, at a particular coordinate on chromosome 12, on a particular gene, I can now change a G to a C if I want to. And this is in a very precise way. This allows us then to build new animal models of a particular human disease. This has not been possible before, and is really a major breakthrough in allowing us to study the pathophysiology of these particular disorders. In this particular gene, as is true of many genes in the brain, mice and man have quite a few differences, but for fundamental genes that are involved in the structure of the synapse are very similar. In this gene, which is called IQ-SEC2, the human and mouse gene are 99.96% identical, so quite similar. A few changes, but essentially very, very similar. The mutation that occurred in the child, we're now able to make the exact same mutation in mice using this CRISPR technology. And we're able to study how these mice behave. So what I've just described for you, the stem cells, as well as this CRISPR-generated mouse model, is a revolution in having platforms, models of human disease in which we can use and apply the idea of personalized treatment. What can be more personalized than an approach to his or her disease based on their particular mutation? And again, this has not been possible until the last couple of years. So let me talk to you about, I mentioned a little bit with the stem cells. I should mention, we can also use CRISPR with the stem cells to actually change the mutation back to being, quote, normal. We get rid of the mutation. And we can see if the changes that we saw in these neurons go away, which again help to reinforce the concept that this gene is the one that's responsible for the disorder. And that's what we've done. We've been able to change the mutation back in his cells that we grow in a petri dish, and see that they no longer have this hyper-excitation. They're no longer discharging spontaneously. The connections that they build between each other are more normal. The mice that we're working with, we've been able to also show that they, as I said, mice are limited in what we can say, but been fortunate to work with groups that have very defined models of social interactions in mice, as well as their ability to remember things. Mice also can have seizures. So probably the most easiest thing for us to see initially was that the mice were having seizures similar to the child. The child was having, as I said, about 100 to 200 a day. These mice, early in development, started developing seizures similar to what the child was having. And we were able to actually do what we call EEGs to measure the seizure activity in these mice. The second aspect of his disorder we're able to use the mice for is to study his ability to remember things. I'll give you one simple paradigm that we've used. And we have the same ability to remember. So let's say we put a mouse in a chamber, and we have two objects, okay? Identical objects. The mouse will inspect these and find them quite interesting. And then I move the position of one of the objects. Mice like people, like things that are new. They like novelty. So a mouse that has good memory will now become more interested in the object that has moved to the new location. And he will have what we say interest in the novel object location. This mutation results in a loss of that ability. The mouse now has no preference for the where the previous object was. He doesn't remember where the object was previously. This is a sign of a cognitive deficiency. We have other cognitive tests as well that show that these particular mice have a problem in their learning of new things. They don't recall things after as little as 30 minutes. And again, this is similar to what we believe is going on with human learning as well. And
Can
finally,
I interrupt you?
Sure.
Sorry, I was going to interrupt you because we've got about 20 minutes left.
Okay.
And there'll be time for questions. So if you want to say the last few things and then we go over to questions.
Okay. So the last thing I want to mention is that, you know, we can use this as a platforms for looking for drugs. But one of the most exciting things that, you know, again, as a physician, I like to take things from the bedside and use them then to try to understand in the laboratory. The child demonstrated something fascinating. This has not been seen for many other disorders, but we're exploring it now for things ranging from neurodevelopmental disease to even Alzheimer's disease. That the child, when he had a prolonged fever, his seizures stopped and he having more improved social interactions. We've been able to recapitulate this in our mice. The mice stop having seizures when we warm them. They basically put them in the incubators that we raise their body temperature to about 38 degrees. And they actually start interacting in an improved fashion. Mice, believe it or not, actually speak to one another. They have improved ability to speak to one another, what we call vocalizations. And this, we believe, has important ramifications for what we know the social behavioral abnormalities that are occurring in ASD. We think that we've made important strides, and our focus is entirely on trying to translate this to helping children with this disorder and other disorders. As I said, I'm in contact with literally hundreds of parents, and each child is going to require this individual, precise approach to their particular disorder, and we hope we can help them. Okay, I think.
Andrew, that was absolutely not only fascinating, but I think you've clarified it down to a level that even those of us who went to uni a while ago can still remember. You know, made it clear. Now, we do have just a couple of questions. Maybe I can even answer some of them. As you pointed out, it's a very heterogeneous situation, meaning that there's multiple demonstrations, multiple causes, and we're not looking at the same gene for everything. Just as you said, breast cancer, when I started medicine, breast cancer was a cancer that occurred in breasts. It is now detailed in many different ways. Anybody who's had it, and I'm sure with the numbers as we know, there'll be many listening to us today, and we all want to know exactly which kind of breast cancer we've got. Is it HR positive, ER positive, and so on? So these are all obviously very different genetics. And the same with the ASD, the ADD, it's going to be quite different. You're talking in this case about a very disabled child. We also know that there are some advantages to having ADD and ASD. They're quite, I mean, a lot of our artistic people have got ADD. A lot of our scientists are on the spectrum. Right. These are evolutionarily advantageous to have. So we're not talking about that. It is a spectrum, and it is, as is pointed out, a neurodivergence rather than a disease. But there are, of course, those with serious illness that need help. Before I throw it back to you, I was asked if it's there's a greater increase in consanguineous in intermarried communities, and I imagine that like every other genetic disease would be true. Yes.
Yes. Yes.
Yep.
Certainly, like you said, Ruth. You know,
Yep.
first degree relatives. There's a much higher incidence of virtually all genetic disorders.
Okay. So that gets us back. I'm not sure that we have any. Can we add some cells back into humans in order to treat the ASD and the seizures? And what about grown adults? Is it different with children?
Okay, this is it's a great question, and it's very exciting. The idea of cell transplantation for certain disorders, yes, it appears. I want to say right yet for ASD, but one application of stem cells, for example, after a heart attack to repair damaged heart tissue. For ASD, I am skeptical that replacing the cells. What I do think might be possible within the next 10 to 15 years is actually trying to do gene therapy. If we identify the gene that is abnormal. Right now, that's not possible for neurodevelopmental disorders. One of the limitations in the Crispr technology I talked about is that it requires that a cell be dividing. And, unfortunately, our brain cells stop dividing very early in development, in our development. It's certainly not occurring in adulthood. What I do think, though, is exciting, and I want to mention a word that some of your audience may have heard about, is something called synaptic plasticity. I think this is one of the more exciting concepts, that our brains are constantly undergoing remodeling. And we talk about ASD. Some of these children, we talk about their wiring may be a little different than others. This is, like you said, a neurodivergence. All of us, though, have the capacity to some degree, this is what learning is essentially, is our synapses, our connections are plastic. By and large, this is greater for children, probably until about age 12 or 13. But we see even in adults, I think a great example is someone who's lost, for example, function of a limb due to a stroke. He can still regain function. He can relearn how to speak, relearn how to use a limb with proper training. So, this concept, we're really at the very beginning of learning how to harness this concept of synaptic plasticity. Very, very exciting. And perhaps through the kind of models that we're building here, we can try to reverse some severe neurodegenerative disorders, as well as help these children that have these developmental delays using this concept.
We have a question. How would you use your recent findings regarding fever to assist children with ASD? I think this is just in a particular situation. Right.
So first of all, I should say that all these things that we're doing, of course, are research, and we certainly are working with physicians around the world. But all these things, anything that I'm saying today, and I'm happy to help any parents that are interested, have to be done under medical supervision with your physician. That said, a very fascinating connection has been associated with ASD and fever. This was first identified at the Kennedy Institute in Baltimore, I believe in around 2006. It had a very large inpatient population. Then an epidemic swept through the institute, and they noticed that the children became much more interactive after and during their fever. And this has been reproduced. The Simons Foundation has sponsored research in this in a number of different settings for children with ASD, and have found not all, but a very significant percentage of children, somewhere in the order of about 25%, do respond with improved social interactions with when they've had fever, and this may last for a period of time after the fever has resolved. We're trying to understand, this is, of course, we think that the relevance of what we found may be helpful to understand what's going on with ASD and other disorders that may be improved by fever And if we can harness the power of fever without necessarily having to cause fever, this, I think, would allow this to become a new treatment.
So you're talking about fever, but you're not talking about just raising body temperature. So sauna is not the same as fever.
Excellent question again. So there's a couple of ways to raise the body temperature. We believe actually the mechanism has to do with actually a raising of the body core temperature. As all of you all know, our body temperature is tightly regulated between around 36 to, you can tolerate okay until about 40. Once you get about 40, a person is really putting a person in danger. Above 42 is virtually life-threatening. So, but what we believe, there's a mechanism in our cells that occurs from bacteria all the way up to humans of what we call molecular chaperones. What is a chaperone? We think about, for our children that are going to the prom, someone that's kind of like going to watch over to make something bad happens. We have molecular chaperones that their function is to refold proteins that are mutated and help them function more normally. These molecular chaperones are induced when the temperature is increased. This, again, we see this from bacteria all the way up to man. Our laboratory is intensively studying specific such chaperones that appear to be able to cause this changed protein, this mutated protein, to fold more normally. And again, this paradigm is going to be true for many other mutations. It's going to help children or adults potentially, that this may be how fever can help a number of different disorders. There's a couple of ways, though, to raise the body temperature. One is, like you said, sauna. Another mechanism that we've actually exploited is to use simply a hot bath, like a jacuzzi. This is a very increasing surface area exposure. Other ways that have been used and are used actually clinically, this is usually a clinical use of trying to raise body temperature, is by injecting certain substances, certain lectins, in some cases actually extracts of bacteria to actually raise the body temperature. We're exploring all these different possibilities to see what might produce the best results. Yeah.
I was just wondering myself if it's a difference between the cytokines and things that are produced during illness, or whether it is pure temperature.
Yeah. We believe it's the temperature that, but undoubtedly what goes on with fever and infection certainly is there's additional things going on. But we believe, because we can see it with the simple by raising the temperature using increasing the external environment, that it actually has to do with the temperature.
Well, you would expect then in summer that the behavior might be better.
Yeah, we think that this we're very interested in this.
which begs the question, what's the temperature in Silicon Valley?
and that's a good question. That's a good question. You know,
yeah.
Sorry,
for those who may have missed that, there is quite a larger amount of autism in Silicon Valley, unless I'm mistaken. Okay, there's a lot of questions. What I want to know is, how does your lab actually work? Who's in your lab working? What kind of money does it take to have this work done?
Okay, well, I think that I really, first of all, I'm here at the Technion, and the Technion's built a wonderful laboratory for us. I trained, as Ruth said, in the States, and we have a beautiful laboratory here overlooking the Haifa Bay. We have a very multi-ethnic, religious population. Students from Russia, students from Israel, of course, from the United States, Jews, Arabs, Christians, Muslims. We all work together trying to help people for these particular disorders. The way it works with regard to funding, it's each of these projects is a project in and of itself. Generating these particular models is expensive, on the order of tens of thousands of dollars. to generate a particular mouse model as well as the stem cell model for any child's mutation. All the students are on what we call milgote or scholarships, which also the laboratory has to cover to pay for supporting the students' living expenses, which on average is about $15,000 a year.
So how close do you think you'd be getting to commercializing or trialing trialing therapy?
Well, I think that we're there. Okay, we are now in a state where we're going from the bench to animal models. Once that we have an animal model that we are convinced that a drug is effective, we are now going to move into to human studies. I want to say, though, that the approach to moving into human studies is predicated on two things. One is having sure that whatever treatment you're doing is safe. And what we're building on is repurposing a number of drugs that have already been used for other purposes, but now based on the mechanism that we've identified are going to be effective in helping to ameliorate or correct the defect that we found in our animal model and in our stem cell models. This greatly accelerates the rate at which we can move from the bench to the bedside. What you're going to get in pharma companies is going with new molecules, which of course they're trying to do because they want to make money. This is a much longer process. We're not working that way. We're repurposing drugs that already exist, or in this case with the fever therapy, redesigning, taking advantage of what nature has already developed. I think I answered your question.
Indeed. And Andrew, it's been very enlightening, even for me who knows a little bit about it. So I hope for those of you who aren't medical or have other interests, I hope it's also been enlightening. Ori, I think you wanted to end up.
Yeah, thank you, Ruth. And I join Ruth with thanking Andrew. That was very, very interesting. We really appreciate that. Like Ruth said, there's still a lot of questions, and I'm going to put you on the spot that I might collect them and send them through to you so we can get back to some of those people. And yeah, there is a lot of very, very interesting discussion, and we might put you on the spot and invite you again in a year to see where the development is with one of your projects. That as you are aware, Technion Australia is supporting a project that you're working on with relation to autism, as well as your colleague, Professor Derry Dickman, who is running interesting research into Alzheimer's disease. I wanted to take this opportunity to thank a lot of our viewers today who already supported us in this project. And also, if you're interested to support us, then I'm going to share a link in the chat, and we'll also send you a link later on. If you choose to support us until the end of this week, we'll match every donation dollar for dollar. So enabling Andrew and his team to do more with your supports. Just two quick notifications. We've got two events coming up. One is with Professor Michael Wallach. Some of you met him. He has spoken for us before. He's a professor at UTS and a visiting professor at Technion, and he's an expert on infectious diseases, and he's going to share insight into how Israel is dealing with COVID-19 versus Australia, and a bit of insight into how the future is going to look. So we'll send you some information about that. And we've got another event coming up early October, which is all around sustainable design and green buildings and the impact on our environment. So on that note, it's 8:01 and we like to finish on time. We thank you all very much for joining. Andrew, thank you. And Ruth, thank you as well. Take care. Bye-bye.